The topic of his talk is Superseding the Synaptic Network, How Cellular Complexity Transcends the Digital Neuron. May I request to Brian J. Ford to proceed for his talk.
Greetings to you all. It's a great honor and a pleasure to be back. We've just heard from Professor Dennis Noble, a much admired friend, about Cartesian philosophy. And in many ways, present-day science is languishing under this bizarre legacy. Descartes believed firmly that everything was a machine. He believed implicitly that if you broke down organisms into their component parts, you would very easily see how they functioned, how they were going to function, and why they were going to function in the way that they did. He was wrong, but the legacy lives on. I'm going to address today a fundamental paradox and a basic mistake that current neuroscience makes in looking at the cell. Neuroscience is quite convinced that the brain is some sort of super organic, amazing computer, and as such will be eventually amenable to emulation by something that humans have created. That's fundamentally wrong. The brain is not a computer. Nothing in the body is a computer. Nothing in the body works like a computer, responds like a computer, or thinks like a computer. But if you insist on an analogy, then it is the neuron that is the computer and not the brain. The neuron, often construed as a kind of organic transistor, is nothing of the sort. The neuron is what thinks. The individual neuron makes up its mind. Neurons are incredibly complicated organisms that nobody has begun to probe. We still think that thought, cognition, emotions and the rest exist at the interstices between neurons at the synaptic connections where they meet but do not touch. And that's not true. The synapses are no more than road junctions on a map. And you may study the road junctions as much as you wish, but that's never going to tell you anything about the sociology of the people who live in the town. People misunderstand microbes. These are bacteria. It is quite possible that this is the first time any of you, distinguished biologists, and if I can call you that, parabiologists that you are, it is quite likely this is the first time you have ever seen living microbes. A film taken by my friend in Canada, Bob Burden, and it showed spiral organisms. This, however, and this is an example from Poland, is how people think bacteria are.
Dožívajte, Brock. Užívajte si baktérie. Jasne, šéfe. Oni si myslia, že nás to zabíja. My sa rozmnožujeme. Čo to tu vonia? Je to tak... Osviežujúce!
Čože? Nový domestos Bhakti stop!
Nikdy som nič také osviežujúce necítil!
Vyblázni, veď nás to zabíja!
Nový domestos Bhakti stop likviduje baktérie a zároveň osviežuje.
No, that's not what bacteria are like. If you want to see a living cell, let's have a look at this one. This is a dinoflagellate alga called Serratium tripos. Look how it unravels the flagella with which it's going to swim. How there are two flagella, intimately intertwined. How slowly and methodically they are unravelled so the organism can swim any way that it wants to go. Or let's have a look at this little organism, Euplotes. It's a ciliated microbe. This is the kind of living cell you've never seen before, and what's more, nor has the public. But do you see how it isn't just wafting food into its gullet, but it's selecting which ones it wants and rejecting those that it doesn't? We are still convinced that physics has the answer to everything. Here is a perfectly typical random example I chose. And it says, as you can see quite clearly, researchers hope eventually to develop basic equations that describe all of life. That is a bizarre overstatement. And the first thing I want us to address is the fact that we don't understand what microbes look like. And the second thing I want us to address is this bizarre belief that in the post-Cartesian world, it's all basically physics. One of the ways in which we've made it possible to address this naive notion is to simplify cells so they become jelly beans. I've spoken on this at this meeting in the past. Here is a more recent example. Cells are just created, models of cells, pictures. They're nothing like real cells. They don't have any relationship to real cells. And as I think you know, I protest about this quite regularly. In regular academic presentations, cells are willfully misrepresented.
If scientists succeed in uncovering all the secrets of the cell, it can be a tremendous benefit for all humanity. We need to look closely at the structure of the cell. Cells are surrounded by a thin membrane of fat. Inside, they are filled with liquid, containing, among other things, nutrients and proteins. Cell research is crucial to understanding how life works. Cells are like tiny machines that need energy to function. The possibilities are enormous as scientists continue learning more and more about these building blocks of life. For the cell's life is part of our life.
Now, this Wallenberg Foundation sponsored research is bizarre. That is not a living cell. These aren't cells either. This is a post-Cartesian cell where everything is mathematics, where everything is engineering, where it's based on physics. And if you look at higher education treatises online, you will find exactly the same incredible imitations. Not looking at real cells, but looking at what people have invented to replace real cells.
Cellular processes can start or speed up or slow down, often leading to complex chains of events that make up the important processes in our biology. The cardiac gap junction and the natural killer cell examples both depend on the cells touching each other. But what if you wanted to talk to cells a little farther away? So paracrine signaling is a good way of communicating with nearby cells, but they can use some of the same mechanisms to talk to themselves. In the next episode, we'll discover how our immune cells learned how to fight off invading pathogens and how they never forget.
This postgraduate course is nonsense. Here's their picture. It's invented random computer graphics, nothing like a cell. I may say that others have done it too. My publishers once put a random blob on the cover of one of my books about microscopes published 50 years ago. So I'm not pointing too many fingers in a disparaging way. We have all fallen victim to it. I mean, here's an example of what a cell is like. It'll tell you about its histones, the mRNA, its spatial position, proteins on the cell surface. And there is that implication that is really a mechanical device that we can very easily manage in mechanical terms, in digital terms, to emulate. If we go online, let's go and have a look for living cell. No, hardly any of these have any relationship to living cells. They're computer graphics. They're simplified computer graphics. They're models we've made up to convince ourselves that we know what cells are like, because we don't. Under the transmission electron microscope, this is what a cell looks like. We've sectioned it here. And in the middle is the nucleus with its dark nucleolus. And there you can see the cell wall. This was done at Radboud University in the Netherlands. But of course, it has a disadvantage. This cell is dead, it's been sectioned, and it's not in colour. Look at a real cell. This is a picture by William Danielson at Wellcome Institute, and you begin to get some sense when fluorescent dyes are used to display specific components of the cellular mechanism. You get some vague impression of its complexity. Cells are actually wonderfully complex things, and although we're always told you need a microscope to see them, you don't. Take a look at this Tradescantia plant. The staminal hairs that cluster around the stamens in the middle of the bloom, if you look very closely, you can see that they look like beads on a necklace. Each of those beads is a single cell, and you can see it with the naked eye. Under the microscope, this is what one of those cells looks like, and you can see the nucleus in the center and the streaming cytoplasm that is busily conducting the affairs of the cell. In some of these cells, the nucleus patrols around the cell as though keeping an eye on what's happening. We don't know why. We don't know how. We have no idea what it's doing. It just does it, and nobody asks the questions. In school, we were taught of amoeba as the shapeless blob of jelly. Well, just look at this shapeless blob of jelly. It's composed of almost entirely water-soluble components, but it's not dissolving away into the pond. Quite remarkable, is it not? And if we look very closely, you can quite easily see that it's filled with a myriad tiny subcellular components. These little organelles all have a purpose, they all know what they're doing, and there's nothing mechanical about it. The most famous picture of a white cell, a blood white cell, which is an amoeba, was taken in 1959 by David Rogers at Vanderbilt University. That's an extraordinary thing. See how that little cell is chasing those bacteria, can sense where they're going, and eventually it rounds on them, devours them, and liquidizes them. And people say, are cells sentient? Of course they're sentient. We've known since 1959 that this cell knew where the bacteria were and knew how to chase them. On the BBC, you'll find a whole raft of selections where you can look at any form of living life you want, and it goes right down to starfish, polychaete worms, and ends up at the Clitolata, the earthworms. It says nothing at all about microbes and the majesty of the single cell.
When I first started out, there was so much of the natural world we were unable to film. The cameras, the lighting, the size of the equipment, all meant that the extremely small, the very large, the ultra-fast, or the infinitesimally slow were beyond our reach. In the last 30 years, things have changed out of all recognition. We can now film everything, from the giants of the world's oceans to the smallest invertebrates.
Now that smallest invertebrate, here we are, it's a leafhopper. It's composed of 5 million cells. You never find people looking at single cells, though in a recent BBC programme I thought we were going to see living cells. It was made by a great friend of mine, wonderful man, George McGavin. If ever you have a chance to see him lecture, do go and see him. This is the result that the BBC came up with.
And then what we can do is we can just wash that out into a cup, and we can collect that to look at under the microscope later.
To actually see them, we're going to have to push our microscope camera to its very limits. What I really want to see, Emilian, is the phytoplankton.
Maybe we can zoom in on this one right here.
Oh, that's nice.
Yeah, they look like some diatoms. Oh, there he is.
Oh, that is beautiful. Fantastic.
Now they managed to pick out just one, but this appalling BBC programme failed to capture microbes, and the closing sequence here is actually 3 separate videos of particles of dirt laid over each other to make it look good. In fact, plankton is so easy to see. Just take a little piece of weed from a pond or from the ocean, and there you will find plankton, little water fleas. There are many cells, of course. There it is. You can see its eye. It has a single eye, which is why it's called Cyclops. And inside we can begin to glimpse some of the majesty of what goes on. And look at its jaw. If you saw a photograph of that, you wouldn't think it was a microscopic organism's jaw. It looks like the jaw of a dinosaur or some sort of massive monster from the past. If we look very closely, those smallest particles there are bacteria. You can see the incredibly intricate structure that the cells within this organism have created. And look at its claws. Are they the claws of a bear or something of the sort? You might think so, but no. These are the microscopic claws of a tiny little organism. Looking at single-celled creatures, like these phytoplankton, which George McGavin hoped he was going to see, and you do get some sense of the beauty, the inherent complexity of these organisms. Here is one that you may know yourselves. It's called Noctiluca, and it is the organism in the ocean which fluoresces at night. It glows when interfered with. I've sometimes swum in waters where Noctiluca exists, and if you open your eyes, you have little sparks of light banging against the cornea. They capture organisms with that single little arm that they have. And you'll see inside this one, there is a tiny little creature, a little embryonic water flea that it's captured, and is trying hard to escape, though I fear it's not going to succeed. In terms of complexity, what about coleps, the great scavenger of fresh water? These are single-celled organisms. Look at the discrete patterning, the delicate patterning, the incredibly intricate patterning that characterizes the cell wall. Look at how it manages to adapt. This is a single cell, ladies and gentlemen. It's nothing complicated. It is a single cell. But look how it manages to find its food. And if you look very closely at the side of the cell, lower down in this view, and you can see very clearly how the little cilia that have been projected from the cell, they're acting like little limbs. They're sensing where it should be. They're changing its position. This is no easy to understand machine. This is an incredibly complicated, autonomous living organism made of one single cell. But nobody's bothering to look at it at all. This is Stomatia, a ciliated organism. You'd never think that that could possibly be a single cell. But look at what goes on inside it. Now you can see why I so resent the jelly bean model of cells, the simplified computer graphic version of cells. This is what cells are really like. Some of you, all of you I hope, may remember paramecium from school, a single-celled organism. Again, you can see the pellicle, the patterning on its cell walls. You can see how the cilia are beating through its gullet as they draw food particles in. And of course, like all animals living in water, the organism takes in more water than it needs, and it therefore needs to excrete the excess water. And if we look closely, this is under time lapse, the last picture was in real time, you can see this contractile vacuole fed by these beautiful little channels that draw the excess water out of the cell and deposit it very neatly into the environment. This is Vorticella, an organism that is important to us because the single cells of Vorticella comb bacteria out of water. And the reason that mountain river water is pure is not because of a lack of microbes, but because of the presence of microbes like these, which have a ring of cilia around the open end of the cell, and they pick up any bacteria and prevent difficulties. Now you might think to yourself, but these cells are rooted to the ground on their little arms, and yes they are, but they can break free. Then the ring of cilia, instead of feeding it, becomes its means of transportation, and off they swim. This is a telotroch or telotroch phase of the organism's lifestyle. And here, named after its trumpet shape, is stentor feeding. And you can see very clearly, if we look closely, that stentor discriminates what it wants to eat. It doesn't just waft everything into its mouth, as most of the textbooks will tell you, but it detects what it wants, and, like Eupliotes I showed you earlier, it rejects what it doesn't. Nobody ever considers the complexity of the cell. Stentor, for example, can also break free and then use its ring of cilia to propel itself along through the water instead of to feed. And look at the complexity inside. This is no simple little transistor. Immense, complicated, multifarious tasks are going on inside. This is blepharisma. And I ask you to speculate on how anybody is going digitally to model each single particle and what it's going to do. I dare say a quantum computer expert will say, well, one day we'll do it and possibly we may. But that's not what I'm saying. What I'm saying is that we haven't done it now. And that is the great lie that's told, that we understand cells. We don't. We don't even look for understanding. Metopus, for example, this interesting little cell, another one photographed by Bob in Canada. Just look at its incredibly intricate structure. What we need to be doing, if we're going to look at neurons, is to look inside the neuron to see how complicated it is. But no one ever does. They look at the synapses, the synaptic connections between neurons. Nobody ever looks to see what's actually inside. And what they do look at, they mostly get wrong. People don't understand how complicated cells can be.
Does it bring up philosophical questions in you? This game's called Spore. I've been working on it for several years. It's getting pretty close to finish now. And you actually start this game kind of in a drop of water. So here we are at a very kind of microscopic scale, swimming around. And I actually realized that cells don't have eyes, but it helps to make it cute.
That's interesting, isn't it? That's Will Wright, a great games inventor at Electronic Arts, who invented this little game. And he says he knows that cells don't have eyes, but he put eyes on to make it look kind of cute. This is his version of a cell. Of course cells have eyes. Not all of them, but many do. This, photographed by Hilda Cantor-Lund, this is Uroglena, a clump of little algal cells. Look at it closely under the microscope and you can see that little red dot at the top. That red dot is its eye spot. And if we section the cell and put it under the electron microscope, you see a curved retina. Yes, cells see. Here is a little red eye spot in Ficus. Beautiful little organism. And what about this? Here is Eutreptiella, a euglenoid organism, one of the Euglena family, and it spends all its time with this bolus of cytoplasm migrating from one end of the cell towards the anterior end of the cell. We don't know why, we don't know how, and nobody's bothering to find out. Here's another one, Euglena itself. You see its little red eye spot? No, this is no simple little bludgeoning organism bashing into its environment in an automatic way. It's a thoughtful, autonomous, considered little cell. Here is one photographed by my late colleague Jeremy Pickett-Heaps in Australia, photographed in close-up. And you can see the incredible complexity of its cell wall. And look, there's its red eye spot, and there is its cytostome, its mouth. If you showed this picture to a child of 10 and said, what is it? He'd say, that's some sort of strange monster. I can see its head, I can see its body, I can see its back end. Never occurred to him that all that might be within a single cell. And yet we still think that a neuron simply fires or doesn't fire a signal like a transistor. Here's one of these euglenoid cells in close-up and held in firm position. And look at the way that it's using its flagellum. Now these flagella are used in ways that we often rarely comprehend. Many of these euglenoid cells live in little spherical groups. And if you look, you can see how this one is selecting what it wants. It's not going to take everything, but look at the one at the top. It's sent out a loop of cytoplasm. It is like, of course this thing is sentient. It has sent out a loop of cytoplasm in order to trap the food that it wants. And it then, in ways we don't begin to comprehend, draws them back into the cell and absorbs it into itself. And that's what white cells do in the human body. Here, for example, is an organism called leishmania. Causes leishmaniasis in humans. There's a white cell, a huge white cell in the middle. And look what the white cell has done to the organism on the right. Now look at what it's going to do to the organism on the left. It's trapped it. It dissolves it. And it will actually absorb its cytoplasm into itself. That parasite is being fed upon by the white cell that has now engulfed it. Here are a couple of these parasites on the edge of a white cell being drawn in, digested, broken down, and actually fed upon. This is happening to you inside your body all the time with any pathogens you may have breathed in. There's 10 of these leishmania cells around a white cell. And just watch how one by one the white cell manages to entrap them, hold on to them. It manages to absorb them, break them down, and take the protein into itself for its own benefit. Many amoebae feed on actively swimming protozoa, which is quite a mean feat when you think of it. Here is an amoeba crawling along at real speed, and when it comes across one of these little swimming cells, it manages to engulf it. Here is a ciliate swimming along. It ignores that diatom above it, which it considers unsuitable for lunch, but then, and we don't know how, it detects another ciliate and wolfs it down. It's like watching a cat catching a mouse, but this is a single-celled organism. Here is a beautiful little alga with a skeleton made of silica. It makes a skeleton of glass. Dissolve away the rest of the cell, and that's what it creates. These little diatoms make beautifully shaped skeletons, and you can find great deposits of them. These are the White Cliffs of California, Lompoc, California, composed of diatomite. Nothing but the silica shells of diatoms from hundreds of millions of years ago. And under the microscope, you can see the perforated little shells. They are, of course, a very mild abrasive in this form and are often used in organic toothpaste. Here's an example. I featured it only because I just love the trade name. I think toothpaste is truly inspired. Here are some algae from a pond. Round ones are desmids. The long ones are diatoms. Diatoms sometimes live in colonies. In this particular kind of diatom, they slide against each other and almost act like an entire organism. So the colony of individual, separate, autonomous cells, by cooperating, can migrate from one part of the pond to another. And here is a desmid, or this sun desmid, very popular with microscopists. What an incredibly complicated cell it is. And it looks just like a disc of chlorophyll. I'm sure that's what physicists would tell you. It's like a disc of chlorophyll gathering solar energy. But let's look under high power and change the focus. And then you begin to see the intricate activity that's going on inside that cell all the time. It's hard to think how they can divide in half. We all know that these single-celled algae must divide. But this, captured under time-lapse by Jeremy in Australia, shows just how the two cells manage to form themselves from one. This is speeded up 25 times. Look at the incredible intricacy that goes on. Now, we've just looked at those round desmids, so let's look at the long, elongated diatoms. And here are two pairs of diatoms and they're going to have sex. If you notice, there are little projections from the two adjacent cells. And the projections, we have no idea how they sense them or how they form them or how they dissolve the cell wall necessary for it to happen. But this, magnified 25 times, allows them to join together and fuse and form a resistant spore that will last throughout the winter. None of those mechanisms are amenable to any understanding, and none of this is taught to students in universities anywhere in the world. You may remember spirogyra from school. Well, here are two filaments, one at the top, one at the bottom, and adjacent cells put out tubes. The textbooks all say they just create a conjugation tube. Nobody has any idea of how they sense that they're near each other, how they sense that the one is the male and the other is the female, how they manage to create these tubes and dissolve the cell wall. But the male filament above migrates, each cell migrates through into the female filament below. And again, the nuclei will undergo divisions and then form a cyst. And then when the winter comes and the cell walls break down, they will survive and hatch out again and form new filaments in the spring. Sometimes the cells don't actually end up opposite another cell, and I'm afraid they don't lose their virginity. You can see there are two cells at the top that never found a mate. And here's another, Oedogonium. This is an alga where the little motile male cell has managed to find its way to the ovum and has identified it, has created a pore, it manages to move its way in. We have no idea how this happens. The entire cell contents migrates within the ovum, and the two of them then fuse together once again to form a spore. This all shows immense ingenuity and senses, and nobody begins to grasp what any of it means or how any of it happens. There's Chlamydomonas. It swims in life with a sort of a breaststroke, and sometimes those two Chlamydomonas organisms will meet, will recognize each other as compatible. They will form a pair, nose to nose as it were, and they will fuse to produce a spore. Here are two that have located a certain synergy between the two of them, and gently those two cells will join together, the cell wall will change, we don't know how, and it will adapt itself, and you end up with a spore. Yes, many of these organisms have sex. Here is, it's almost, I don't know, almost not quite erotic, but it's a very endearing picture of two Blepharisma protozoa that have met, and they are undergoing sexual conjugation. These are single-celled organisms. Nobody has any idea of the intricacy of the mechanisms that go on. And every time we do create a model of a living cell, it's always as though it's plastic or mechanical. Here's an example from the Science Nature page. Here is a plastic model. This is quite a good model as it goes. But the whole point of it is that this is filled with solid blobs or like transistors or components or something. But that's not what cells are like. A real living cell, like this Parahelicea, is in fact filled with amazing complexity. Each one of those organelles is as important as an organ within your body, but it's within a single cell. Just imagine the complexity of what's going on within your neurons. No, they're not just firing off signals. They are complex and autonomous and thoughtful. And yet wherever you go, you find people down-base cells and create simplified versions simply to satisfy the fact that the public then will feel you know a lot, when in fact you know nothing at all.
This lecture hall will be made of code, and though they're virtual students, what they'll learn together is real. The impact will be real.
No, it's not real, Meta. Your metaverse is not real. That is not anything like the way that a living cell divides. And I'm not the only one who thinks so.
Aaron Bernstein, doctor and research associate at the Centre for Health and the Global Environment at Harvard Medical School, has been studying the ties between the natural world and human health.
Really, we're utterly ignorant about the microbial world. In fact, it is the last great unexplored frontier in life.
He's absolutely right. And not just microbes. This is Colpidium, a little colony of Colpidium in a pond. But it's not just microbes, you don't understand. It's single cells as a whole. Here's another ficus. You can see its red eye spot quite clearly there. And how amazing it is that all this goes on inside a cell and yet we know so little of it. Many of these cells have exquisite beauty and some almost bizarre appeal. This is lacrimaria, named from its supposed likeness to a teardrop. And see how it explores its environment. It's like I don't know, an anteater or an elephant, but it's just a single cell. So is this. Facadinium. Each one of those particles is carrying out important tasks, and no one's going to just model that with a simple computer. The idea of modeling a neuron or modeling a microbe is just a fantasy designed to get big grants and confuse the public. Some of you may possibly eat spirulina as a diet aid, and you know that spirulina is a blue-green alga, the simplest kind of alga you can imagine. But under the high-power microscope, you can see that, yes, there's lots going on, even inside that cell. The trouble is that nobody ever understood the way in which the study of the neuron developed. In 2007, I wrote a chapter for this book, Brain, Mind and Machine, and it's the first major publication to look at the way in which the microscope was brought to bear on neurons. There is Leeuwenhoek's microscope. I might add that I have recently been examining under the scanning microscope another previously unknown Leeuwenhoek microscope recently discovered in East Anglia, not far from where I live. These are the specimens and a drawing on the right that Leeuwenhoek did when he studied nerves. He was the first person ever to study nerves. And as the centuries went by, people began to regard nerves as though they were little tubes full of fluid, as though they were mechanical devices. I took a photograph of nerve cells on the right. They're photographed through a single lens microscope of the kind available in the 1600s. And yes, you can see neurons there perfectly clearly. But this man, Purkinje, he was the man who actually put the seal on the study of microbes. And one of the drawings of a single neuron that he produced actually gave rise to the term Purkinje cells, and they're still called Purkinje cells to this day. That's what they look like under the microscope if you take a thin section. But do you notice how many of the side branches, the dendrites that come out of those cells, go out of focus?
That was a problem that caught the attention of this man, Ramon Santiago de Cajal.
In order to overcome this focusing disparity, he wouldn't use any sort of recording other than pen and ink. He would change focus in order to find exactly where those dendrites went, and his drawings have never been bettered. It is astonishing to think that in the Victorian era, this man was showing us so much about what goes on in the world of the neuron. There is a single neuron, and look at all the dendrites. It's unbelievable. And he actually mapped great chunks of the brain and drew neatly every single neuron and the pathways of all of its dendrites and axons. This, if you go to Wikipedia, is how you will see a single neuron. Diagram's all wrong. The middle of the cell, the body of the cell, is filled actually with what are called Nissl granules, which are absent from the axon hillock. You don't see that. And the axon, which is the thing that actually conveys the major message out of the cell, the axon actually should have a whole series of Ranvier nodes. It's not just a cylindrical extension as you see there, but an extension that's interrupted at regular intervals. It's all wrong. Now, this is one of the major points I want to make, is that we falsify what we know for grants. Witness the beauty of one of science's greatest mysteries, the human brain. We're going to have a view of life at a cellular level never before. No, it's completely untrue.
Life of a Neuron takes us through the journey of human growth and the mind, and it really allows us to immerse ourselves in memory and in those experiences. And using sound to trigger memory in an immersive environment such as ELISA allows the audience to really experience both their own past and the story that's being told in front of them.
This Art Tech House production is wonderful. It's absolutely vivid, but it is not what it says. It does not take you through the life of a neuron. No one's ever done that. Nobody knows the life of a neuron. These are pictures, scanning electron microscope pictures, which are then converted into computer art and coloured, and just made into a dazzling array. But they actually tell us nothing of what they say they're telling us. And this is a great lesson in the field, the rampant dishonesty throughout neuroscience. Here, for instance, is a bit of film from UCLA. This is Professor Katsushi Arisaka has taken this film. And it's interesting because it shows how some neurons seem to fire off when others don't. But it's so basic, so primitive. The resolution is so low. We get no impression of what's going on inside the cells. You just see cells flickering on and off. And yet he says that captured using 2-photo calcium imaging, this video depicts neurons firing in the brain of a mouse in response to stimulation of his whiskers. And if you study that, then you may be able to understand why children with autism covered. No, you won't. Got no relationship to autism. And look at this. Here is another example. 5 fluorophores were used by Professor Edward Boyden and his team, and they have shown how 5 coloured dyes at the same time can illuminate bits within cells. That's quite interesting. All you're seeing is cells dyed with a dye, but when they reported it, it comes out once again with massively complicated computer graphics to illustrate the point. And here's another one. Scientists are feeding artificial intelligence chunks of human brains. No, they're not, and it wouldn't matter if they were. This is a paper published in Nature. Scientists grew mini brains from stem cells. No, they didn't. The stem cells, which they pulled out and cultured, still managed to survive, and since they knew they were supposed to be generating eyes, still managed to generate little islets. It's the cells that did it.
Everything you see here is real data. We're looking through a microscope at neural tissue, going down through an area of the brain called the medial prefrontal cortex. We can use these microscopic images to reconstruct the 3-dimensional layout of the neurons and really see the intricate and intertwining patterns that they make. These connectivity patterns throughout the brain underlie all neural function and are responsible for everything from controlling your muscles when you tie your shoes to how you feel about your family. Understanding these patterns is a massive ongoing project for neuroscientists everywhere and could lead to breakthroughs for everything from consciousness to...
No, the brain doesn't control everything. The way that all of the white cells in your body and the cells in your liver and your kidney are working, they are invisible to the brain. They're not concerned with the brain. You don't actually need a brain. There are people whose brains have been irretrievably damaged but who still stay alive. The brain is responsible for your senses. And the Human Brain Project took €1 billion and claims to be a great success. They produced some amazing pictures with these fluorescent dyes and produced booklets and exhibitions to say how successful Europe was. No, they weren't. It's a con. They got $1 billion and published 3,000 papers. And what does it tell us about the way neurons function? Next to nothing. Artificial neurons mimic complex brain abilities. No, there aren't such things as artificial neurons. If you look at a single living cell in culture and you get some sense of what goes on inside and what none of us really begins to grasp. This is a human cell. Look how it's put out ruffling extensions. It's searching for the other cells. It's wondering, why is it here on its own? And then when you look closely, you can see some of the miraculous intensity of what goes on inside the cell. The closer you look, the more you realize that these cells are much more complicated than you think. There's the ruffling. This cell is sensing its environment. And look at the busyness that's going on inside it. No, I'm sorry, I will not accept that people understand neurons because they don't understand the complexity of an autonomous cell. And a neuron, we would all agree, is the most complicated and highly evolved cell in the universe. Then if single cells can do things like this, imagine what the neuron does. We think of mitochondria as being little factories, but no, they actually move around. There's a mitochondrion, and it's jiggling up and down through that microtubule and changing its shape. It knows where it wants to go and what it has to do. So let's, as we end, look at Cellular, a single cell of this remarkable organism. And you had to stop and realize that inside every one of those particles, none of which we understand, is complicated, self-regulated activity. You can't model that, ladies and gentlemen. You've got something which is not a molecular model, not a kind of organic transistor. This is life. And I have to say to all of you that until we understand the need for humility, until we recognize the fact that scientists and neurologists are exaggerating their findings simply for the sake of getting grants out of the organizations that will fund them, then once that happens, I'll show some enjoyment and some relaxation. But until then, I shall keep reminding people that you understand next to nothing And it's not good enough to say we're on the point of curing major diseases through our discoveries. We aren't, because we're not asking the right questions and we aren't making the right discoveries. Thank you.
Thank you very much, Professor Ford, for a very informative, effective, and in-depth explanation.